Welcome to this session of Metabolomics for Dummies. And let me just introduce with a few slides before we go on to Andresa. So first I will do a very quick overview, then there will be an introduction of basic concepts, and we will go into the analytical methods and instrumentation, and then we will see a bit more of the data analysis. So what we do with metabolomics is to, we seek to understand how molecules affect disease. And then, so how many molecules are there in the body? And it's estimated there are as many as 250 million different metabolites in the body that, as you can imagine, these are many. And then we don't have only metabolomics and lipidomics, but we have other omics. Next. And here is just an idea on the other omics and how much they are growing. So as you can see, metabolomics and lipidomics, they're actually growing very much in the last years. And in this presentation today, we're not going to have time to talk about data integration between the omics, but I want to just give you a couple of examples of work that we've done with the integration of the omics. So this is a bit like starting the house by the roof, as we say in Spain, but I just want you to see what happens at the end. So in these two examples, we have a cohort of 240 people, and we had omics, which were lipidomics, and also proteomics, and also some of the genes. And in this case, it's for Alzheimer's disease. So as we had a lot of data, what we did was to do a cluster analysis. And this means that we were able to do groups of proteins and lipids that after we could associate with the genes that are known to be risk of Alzheimer's disease. And like this, we discovered some of the lipids that are interesting to look for Alzheimer's disease in a more causal manner. Next. And then with the same data, we did another analysis. And this one is what we call more granular, because now we use particular proteins and lipids. And here we manage data reduction by using an algorithm, which is machine learning, Lasso, which actually finds the most important features for your analysis. And then what we did is a network where we have here in yellow Alzheimer's disease. And then we have clinical data that is important for the disease. So here we have very near age. And then we have a network of proteins, and then how these proteins are talking to the lipids. Next. So, and just to finish this introduction, this is the team. And just to say that the Metabolomics actually has a lot of different expertise, and it's really the work of many people. So as you can see here in the lab, we have people that are experts in big data, other experts in biochemistry, analytical chemistry. And today we are lucky to have the introductions by Andresa, and also Carolina and Tommy. And if you want to know more about the publications from the team, you can go to this address over here. And now I'll pass to Andresa to go more from the beginning and more of the basic concepts.
Thank you, Christina, for the introduction. So today I'm going to share with you some basic concepts within metabolomics. So let's start with the definition. The word metabolomics comes from the combination between metabolites with omics. Omics stands for the investigation of a complete class of compounds found in species, while metabolites refer to small molecules with a molecular weight under 1.5 kilodalton that are producing living organisms. All metabolites together compose what we call the metabolome. which is characterized by chemical reactions that produce small molecules that are essential for growth, development, and reproduction. These essential molecules are part of the primary metabolism, and they can be lipids, amino acids, nucleotides, organic acids, and sugars. One example of a metabolite is the glucose, which is essential for our body to obtain energy. You can see the pathway, metabolic pathway, related to glucose. We also have the secondary metabolism, which is responsible for the production of specialized metabolites that they are not essential for keeping the organism alive. However, they may confer an additional advantage for this organism to survive. Here we have an example, the secondary metabolism of plants that produce some metabolites with the bactericide effect that might help the plant to be resistant to pathogens. As you can see, there is an intrinsic relationship between genome, transcriptome, proteome, and metabolome, and the integration between the omics sciences is essential to understand the mechanisms behind diseases, as Christina showed a very good example of the potential of the combination of all the techniques. You can also see the coverage of compounds of each technique. And what are the goals of metabolomics? So the primary goal of metabolomics is to obtain the fingerprinting of samples. This means that we aim to characterize the samples using a large number of variables that represent metabolites or metabolic signatures. Metabolite signatures, by the way, can be represented by chemical properties obtained by analytical techniques such as mass spectrometry and NMR. Another goal of metabolomics is metabolite identification, is finding out metabolites associated to specific organisms or specific to some conditions like diseases. And when you identify metabolites associated to a disease, for example, you can target, you can track the pathways related to these metabolites in order to propose strategies for treatment or even prevention of this disease. And the ultimate goal of metabolomics is to find a metabolite that represents a biomarker. Here is a typical pipeline for metabolomics studies, which starts with sample preparation, then we have data acquisition, and finally, data analysis. The choice of the appropriate steps to be included in these pipelines depends on the biological question. According to your biological question, you can then design what type of data you are going to acquire for your metabolomics study. So you probably heard a lot about target data, semi-target or semi-quantitative data, but what exactly do these two mean? So I will start to clarify the differences between targeted and untargeted analysis in metabolomics studies. So imagine that you want to investigate a completely new disease, that you have no clue about the metabolite signatures associated to this disease. There is nothing in the literature. And if you find out more about this, you can win a Nobel Prize. So in order to increase the probability of finding metabolites associated to this condition, you need to have a broad window of investigation in order to screen as many metabolites as you can. So when you use this qualitative or exploratory analysis without knowing exactly which metabolites you are looking for, we say that we are doing untargeted analysis. On the other hand, when you already know in advance that a specific disease will affect specific metabolite pathways, and you can then look specifically, monitor exclusively those metabolites that you know that they are affected with a lot of details in a way that you are able to also quantify these changes in metabolite profiles, then we say that we have targeted analysis. To do targeted analysis, you really have to close your window of investigation and focus in a specific class of compounds or a specific panel of compounds. And when you do this, you lose information of the overall. However, you gain lots of information about the details. And this can be very useful because it saves a lot of time. So what should you analyze? Something that is not relevant for investigating this disease. So we say that on-target analysis is the very first step for biomarker discovery in metabolomics studies, specifically because you can screen a large number of compounds. So in the laboratory settings, what are the differences between targeted and untargeted analysis? To do targeted analysis or quantitative analysis, you really need to have references which are identical to the compounds of interest for every single molecule that you are monitoring. In mass spectrometry, the most efficient way of doing this is to spike your samples with heavy label isotopes at known concentrations in order to obtain a ratio between the response of your compound of interest in relation to the response of your internal reference. For doing quantitative analysis, we need to have calibration curves with external standards identical to the compounds that you are monitoring. In untargeted analysis, you also have some internal references. However, you don't have for every single compound that you are monitoring, but you have references for classes of compounds of interest. And in untargeted analysis, you also don't have calibration curves. What you have is intensities of signals. You're not able to obtain absolute concentrations like you do for quantitative analysis. And there is also an intermediary type of analysis between targeted and untargeted that we call semi-quantitative. And I'm not going to discuss this in details because it's taking too long. But if you have questions, you can ask me afterwards. Okay, so here we can have an overview summarizing all the type of metabolomics approaches that we can use in a study. And then you can use this as a reference to decide which type of analysis you can use in your study. So if you want the amount of compounds that you want to screen is the most relevant information for your study, you should definitely go for untargeted analysis. On the other hand, when you already know metabolites that they are changing in your system, then you can use target approach. So what are the advantages of using untargeted metabolomics? First is that you are able to acquire all possible signals of unknowns. It's also your best chance to finding a biomarker. And you can also do semi-quantification with untargeted data. However, the data is very noisy. It's a three-dimensional data that is very hard to do data processing and post-process. For targeted metabolomics, the advantages is that you can obtain absolute concentration of compounds. This is really interesting, especially for clinical research, but also for other fields. This type of data can be easily incorporated with other omic approaches, and the acquisition is really fast. The downsides of targeted analysis is that you really need to know in advance which metabolites you are looking for. And also is a very expensive type of analysis because heavy label isotopes have a very high price. And in fact, sometimes you can even find available commercially some of the internal standards that you want. So having all of this information, you can then decide which type of data acquisition you are going to use. Then the next step is to choose the instruments that you are going to use for metabolomics studies. And now I'm opening the floor to Carolina to talk more about instrumentation in metabolomics.
Thank you so much, Andressa, for a very nice introduction. So we are here now. We're going to talk about the technology and the methods, and I will show you actually what is specific for the very nice setup at Stino. But let's take a little like a mental break, and I would like to introduce you to a molecule that saves lives every day. It actually helps doctors to do their jobs every day, and basically a lot of things in general in the world would not happen without this molecule. Um, in normal circumstances, I would ask you to try and guess what it is. But since it's on Zoom today, I will just tell you that this is caffeine. So with the caffeine, with the coffee, if we think about it, just the coffee is not just a coffee. It's actually a mixture of many different metabolites with aromatic compounds and other type of small molecules. Um, so let's just quickly have a look, quick look at the chocolate, what's happening when we're eating chocolate. There's different types of molecules with many different structures and masses. And not only that the chocolate, what we eat, gives the molecules to the body, but actually the response from the body itself, let's say we're getting happy, the serotonin levels are increasing. This all to show you that actually every day we produce, metabolize, burn, make, inhale, exhale, intake, not even excluding the gut microbiota itself, a lot of different compounds, starting from the cellular level through um, through the tissue, looking into entire body, external, internal factors including. So this actually gets me to the point that metabolomics is extremely diverse field. If you think about it, compare, I think, to other type of omics. And therefore, the analysis of them requires many different approaches. They vary a lot in the size. They vary a lot in the hydrophobic hydrophilic properties and the volatility by themselves. Also, the basic separation in metabolomics, I would say, is between type of polarity. That's the basic one. The polar that are sugars and biogenic amides, ketones, amino acids, and here you can see an example of, let's say, Krebs cycle, and apolar lipidomics. And as Christina said, lipidomics is one of the most increasing field. It's because it's actually a bit easier to identify them because of the structures and predictable because of their predictable structures, but also the presence of lipids are actually extremely abundant. We're looking at every cell in the body. We're talking about the membrane. We are talking about different types of foods, and of course the lipids that are causing damage to the body. That includes, for example, the NASH and different liver problems, as well as obesity and type 2 diabetes itself. I said that the diversity of the metabolites, it's causing the need of different analytical platforms and different analytical needs to actually try and quantify as many of them as possible. In our group at Stino, there are many different platforms, but in our group, we specialize in mass spectrometry, liquid chromatography, and gas chromatography. But mass spectrometry is the key one that we use here. What is mass spectrometry? Actually, as it says, it measures mass, but it's not really only the mass of the molecule. We actually measure mass to charge ratio. And what does it mean? Why is it mass to charge and what not just mass? So this is how when you open the file and you have a look at the mass of a specific molecule, you have a look that the mass is 652.36. I know, not extremely impressive, even if you think about the laser microscopy or something, but actually this provides quite a lot of nice information. So what is happening is that if we look the ionization part, is that we are injecting the sample in a liquid form to, let's say, liquid form for LC, liquid chromatography. We're ejecting the sample and it's being ionized. And ionized, it means that to a specific molecule, a charge is attached. And depending on the properties of this molecule, we get different stages of charge. Most of the charge that we experience in metabolomics is +1 hydrogen or -1 hydrogen. So it's positive or negative mode, which means that if you look at the mass, you have to add or deduct the mass of the proton, and this is your mass to charge ratio. To have a very quick look, these actually molecules which are being ionized, we are able to detect them using as many different types of detectors that are inbuilt in the machines. So let's say there are different type of detectors, and two examples of them are, for example, the thermospecific Orbitrap or Bruker time of flight. And how the ions are detected, it's inbuilt, but but actually we also have to think about that each design comes with different properties in terms of sensitivity, which means that when I say sensitivity, how low in the concentration or in the abundance of the molecule we can go. Resolution, which means that how well I can see difference between two compounds of almost identical mass to charge ratio. And scanning speed, which means that if I have a mix of different ions coming in at the same time, how well the machine can process this. The concept of mass spectrometry is very closely related with tandem mass spectrometry, as I would call it MSMS. The idea is that if you think about it, one mass to charge or one mass can correspond to many different structures, which means that you kind of have to try and figure out what is the real structure and not just the mass. We are using for this a collision, and this is MSMS. So which means that we have one molecule in MS1 measured, it goes into collision cells, and through MS2 analysis, the fragments are being detected. It's like you have a puzzle that they look on the same, and you have to break it and put it into pieces, because those pieces will tell you much more than just one single mass. And this brings me how actually we identify the metabolites. Because as Andresa said, we have two ways of analyzing molecules. We have the untargeted and targeted metabolomics. And in untargeted, we're going to have a lot of unknowns. But we can identify some of them using so-called spectral libraries. So we have a molecule, we have a mass spec, that is the green one, that we say in the library, we match it to what is in the libraries. And these libraries can be public. These libraries can be something that you build, destroying all sorts of known molecules. So we match it later. We see if it's this one, if it's matching this one, or this one, or this one. And then it's like, okay, it's a perfect fit with a certain percentage. So it must be this molecule. And yeah, and basically this is how you identify, and you have to then understand that this is quite, in terms of untargeted metabolomics, it takes quite a bit of time and quite a lot of resources to do this kind of analysis. And still keep in mind that when you get to your raw data, there will be from 1,000 to few thousand we call it molecular features, so the unknowns, and perhaps 5 to 10% of identified ones. And why? It's because a lot of them we still don't know. There is a lot of possibilities for discoveries, which is, I think, so fascinating about untargeted metabolomics. But looking into chromatography, we have to separate the soup. And I say the soup because if you have your little sample, you extracted the metabolites, but it's one sample with all of the mixes of metabolites. Machine, if you look at here at the picture, is not happy if you just feed it like this. It will not be able to process it, which means that we basically have to make a line, a streamline, that each metabolite will go one by one to the machine for being analyzed. And why we do that is because we want to make the metabolites accessible for the MS and MSMS, and we do not want the different types of interference between them. As I said, there's a lot of them, and there's a huge mix of different information, especially in the untargeted one. Chromatography has two ways of separating them. The one we use, at least here at Steno, gas chromatography, which basically pushes the molecule through a very long column using a gas, and depending on the how volatile are the compounds and how they interact with the surface that is on the column, it basically creates a retention time. And some of them retain, some of them go very fast. The same principle is in liquid chromatography when we use different type of phases, which means we do different organic solvents. And the difference is that we use liquid to push through the molecules and create nice retention, because they also have they interact differently with the column that basically has a very specific features for specific metabolites. For those who never saw a chromatogram, this is how it looks. We have intensity of a peak and we have a retention time. If you look inside in the data, there will be a mass. As Andresa said, it's a 3D type of data for normal MS. At SDCC, we have different type of platforms, and for Galaxy and MicrobLiver, those three ones I'm sure you are very familiar with, and we are currently working on setting up another type of platform that will be used mainly for super fast, super short methods for many thousands of samples. These are our strong working machines. Going a bit into details, our QTOF is measuring approximately 1,000 metabolic features. We can annotate approximately 300 lipids because we mainly use it for lipidomics. These are suitable for different types of lipids, and this is really cool because this method has been validated by interlaboratory study. Yes, and you can see more or less the structure of what kind of data you can see. Next one that is the best for the polar compounds is GCGC MS. This is gas chromatography, measures about 1,000 metabolic features, annotates about 200. We can also quantify here approximately 30. Very suitable for all sorts of different polar compounds, as I said, and very nice to look closer into pathways that are quite well known, but also has the possibility for new discoveries. The last targeted method, as Andresa said, this is something we've been working, the group has been working for quite a while, and constantly adding and constantly developing on top of this different molecules. From now, it's about 30 plus, I would say. This has been a contribution from many different people. And I think from here, I will give a hand when I show you how we analyze it. I will give a hand to Tommy to show him actually that he will show you what we do with the data. Thanks.
So I will present some statistical approaches and workflows for analyzing metabolomics data. So I will start by telling a little bit about pre-processing, the raw data, how that goes into a more simple format. Then I'll talk about modeling approaches, starting with metabolic modeling, which is a more complex approach, going into enrichment analysis of pathways. What does that mean? How does it work? And then finally, I will tell about lipids and the lipidome and how those are special among the metabolites. So first, about processing, as Caroline also showed in some slides, we get from mass spectrometry this kind of overview of data. So we have different masses, compounds of different masses coming through the chromatography at different times, and each of these gray dots in this map are potentially a unique compound. We don't know at this stage, but that's possible. And then we need to crunch these into something meaningful. So yeah, if we look at one single point here, that's basically a scan in time of different masses. And when we integrate this data over time, we get peaks, which represent compounds. So that's the signal. And then we need algorithms to crunch this into an Excel table or this kind of simple format where we have participants and compounds and levels, concentrations of compounds. So this is a rather complex area of science itself. We need algorithms to get this signal into numbers, and we need databases to identify the compounds. What is each of these dots that we don't know to start with? And when we get to this point, we can then start working on the data and ask questions. Are there some metabolites that change in the disease? And so in data analysis, I will start with the metabolic modeling, where the idea really is just to have a map of the phenomenon. So we have the metabolic network that you could consider as a map like this, where we have roads that lead somewhere and cross, intersect each other. And in this map, we have reactions between molecules, the crossroads, and in the crossroads we have substrates and catalysts or enzymes in biology usually, and products, what comes out of the reaction. And then there's a rate of this reaction. So every junction in this network is a sort of a reaction. Something goes in and something goes out. This is also roughly how metabolism works. And then, so that's just the framework, but when we measure data, we get concentrations of this network or the traffic in the network. So what is exactly happening within the roads? And that's basically omics. We measure compounds and we try to find out where in the network there is something happening, something unwanted happening in disease or in treatment, how the network is responding. So one example study of this kind is this paper where the authors looked at alcoholic fatty liver disease by integrating multiple omics, gene expression, proteomics, metabolomics into the metabolic network of liver cells. And by doing that, they identified what is changed in the disease in the network, where the balance is gone wrong, and they could find that certain amino acids are basically shut down, or there's a change in the synthesis of these. So blue arrows happening here. So we have less traffic than what we would expect in a healthy cell. And as a follow-up, this study was taken forward. Like the results first were based on data, but then when the authors knew what is wrong in the network, they did a simulation study that how can we fix the network? So let's fix the roads in a way that there's better flow through the network. And they took some key metabolites of the network, handful of those, and simulated if we bring more of that into the cells, could that fix fatty liver disease? So could the change in the metabolism by bringing in these compounds, more of that, lead to better balance of fat in the liver? And they simulated this, so adding different amounts of these compounds, and eventually found a balance where each of these reactions is in a normal state again. So that was a sort of theoretical way of trying to solve this question. And then once this optimal recipe was figured out, they did a small clinical trial, feeding in this mix of metabolites and see can that actually change the liver phenotype. And yeah, this is not fully published yet, but there is some first results about that in arXiv. But in a very small study of participants, feeding those right metabolites for the participants every day, they could see some changes in the liver health measures. So yeah, some pilot example of how to translate multi-omics results and complicated metabolic networks into something that could work as a treatment. But yeah, I will switch gears then to enrichment analysis, which is basically a simplification of metabolic modeling. So this is, like I said, rather complicated approach, and often we are not really measuring all the compounds in the network. So there are just so many metabolites, like Carolina mentioned, and those cannot really easily be measured, and we need many different methods to do that. And then again, secondly, this is only a snapshot in time. So this might be a network of 4 p.m. traffic, but we all know that it's very different from 9 p.m. when it's a different flow. This is the same thing in disease. Different stages look different, and also in when you take a pill, half an hour later, 2 hours later, or 1 week later, they're different phenotypes. So these kind of variation bring some obstacles to these modeling approaches. So we can simplify it by enrichment analysis, where we just really look at one neighborhood at a time. So we don't need to know the reactions exactly, and like what goes in and what goes out, but we know that these metabolites are acting together in some way. And if we measure enough of those, we can figure out is this group of metabolites somehow changed. And this is an example of then pathway category. We have a neighborhood in the map where we see more red than what we expect on the whole map. So we can infer that there's something going on here. There's an enrichment of traffic. That's essentially what we are doing in omics. Also, when we do enrichment analysis, we look at the different parts of the network and find out, is there something unexpected happening? And how is it done in practice? Well, there are like different databases where we can find information about metabolites and other biomolecules. This is an example that also was mentioned in the previous talks, HMDP, Human Metabolome Database, which basically covers most or really many metabolites and biomolecules. And it shows information like this. We have molecule called valine, which is an amino acid, and then we have different information. And we have also information about the pathways that it participates in, about diseases where it's changed, and about like treatments or drugs which activate or deactivate this compound. So for example, here in this page, you can see different pathways that it's involved in. You can click through, see them visually, and understand what is the context. And but when we do it in data analysis, of course, we do it in a more automated way. We don't do it manually, going through everything, but we bring this data into the algorithm and then crunch the numbers, find out are there many molecules in one pathway that are changed. So brief example, here we had also a study on liver fibrosis in rats, where they measure different metabolites after the start of the disease. So we see some changes in the metabolites over time. And when we bring in this pathway information and do the enrichment analysis, we get a result like this, where each square is a rectangle. Sorry, circle is a one pathway, and y-axis shows the significance of that pathway. So we have some valine amino acid pathways changed, as is shown here in these individual metabolites. And then we have a lot of pathways that are not really changed. They are just here around 0. So some individual metabolites might be changing in those two, but not really overall. And the most significant is sphingolipid metabolism, which are lipids that are changing. Here appears to go reduced in the disease. And lipids, they are actually some particular type of compounds. This is a sphingolipid, where it has a head group and fatty acid chains. So there are many lipids of the same kind with the same head, but they are of different size and different saturation. So there are different lengths and different numbers of these unsaturated bonds. And this information you can find on Lipid Maps, which is one database on lipids. So here I'm listing all the sphingomyelins. It's about 100 different ones that have very similar name, but just different numbers. So they are different length and saturation. This one is SM38-1, so it has 38 length and 1 double bond. Yeah, so lipids, it's not so meaningful to study them in the same way as metabolites. They're basically all part of the same pathways, so it's not really very informative. But we can look at them in this way, for example, where we put them, categorize them by size and unsaturation. So these same properties here are on x-axis and y-axis, and we show basically the change in disease in color, and each rectangle is one lipid species. So we see, for example, SM38-1 here in 38 and 1 about here, and that one is blue, so it is reduced in fibrosis at Kleiner stage 4. And we see basically this kind of pattern where larger sphingomyelins are reduced and some smaller ones are increased in the amount in liver disease. This result is from Galaxy, actually, and by doing this more systematically, we can look at many lipid classes like sphingomyelins, as I showed, phosphatidylcholines at different stages of disease. So going from more healthy liver onwards to fibrotic liver. And yeah, this data we have integrated with other omics, gene expression, gene regulation, and also lately like a Mendelian randomization study to validate some of these findings. And these type of analysis you can do with lipidomer, which is actually an R package that I have created. You can install it with this command and find some examples on the web page. So with that, I'll just wrap up. So we need processing of the data to start with, and we can use metabolic models to explain the metabolic networks. And when we measure omics, we can actually understand what is going on in the network, what is changing. Enrichment analysis is basically a simplification of this approach, where we just look at groups of metabolites together and try to see, do they change coherently or not? And then finally, lipids are a bit particular type of metabolites, and those we can look for patterns in size and saturation within each class. And these parameters might be something that is relevant for disease. So thanks for this session. I will just show the acknowledgements in the end before we go to questions and discussion. So this work is of course part of Galaxy and MicrobLiver, which are funded by Horizon 2020 and Novo Nordisk funded.